Anitocaptogene autoleucel is a BCMA targeting CAR that has a similar domain on the inside, but on the outside has this very small binding domain that binds to the plasma cells called the D domain. Because of its small structure, it has some theoretical advantages, including being able to make the CARs in a way that they express more of the grabbing target piece on the outside of the cell, and potentially that you can give a lower cell dose. So the trial that I'm going to present the preliminary results of today is the phase 2 registrational study of anito-cel in patients with relapsed and refractory multiple myeloma. We're going to present the very preliminary and early data cut of these patients that were treated. They have all received 3 prior lines of therapy. They're triple class exposed. They've seen the big drugs that we often use for patients, so immunomodulators like lenalidomide, proteasome inhibitors like bortezomib, and CD38 antibodies like daratumumab or isatuximab. They'll all have seen these 3 major classes of drugs and progressed, and they had to have refractory disease to be enrolled on the study. And what we saw at this early data cut was that the overall response rates of having this CAR T-cell therapy administered to these patients, 97% of patients had a response. And of those patients at this time point, over 60% had a complete response or better. What was even more interesting is that if you look at the depth of those responses, so patients achieving this minimal residual disease negativity, so that's something that we look at increasingly now for patients, how deep those responses are. When we look at a sensitivity of 10 to the minus 5, which is very deep, that many cells have to go through to find just one myeloma cell. Over 92% of those patients achieved MRD negativity of those who had the evaluable test done. What's really interesting is that MRD negativity was achieved at a median of just 1 month from receiving the CARs. So we're seeing these very deep, very durable responses in the phase 1 study that hopefully are going to translate into the phase 2 study as we get more follow-up. Yeah, we're going to present a safety cohort as well. And this is, again, very current, recent data cut. Look at the data. And what we're seeing is that 83% of patients have some kind of CRS, cytokine release syndrome. So that's where patients have this inflammatory response with fevers. 17% of patients had no CRS at all. And of those patients that did have a CRS, the majority were grade 1 and were treated as per protocol and in line with how we would always treat patients with cytokine release syndrome. And 2/3 of those patients that received dexamethasone only had a single dose. At 14 days, nearly all of the events have completely resolved. So that could have a big impact in terms of patients having that side effect completely resolved at 2 weeks post-infusion of CAR. And certainly for the neurotoxicity, which is something that we always worry about, the immune cell-mediated neurotoxicity syndrome, or ICANS, that was observed at a very low rate, so just 9% of patients total, so 91% of patients did not have that occur. All of those events were completely resolved at the time of discharge. And what's interesting is that at the current time point, we haven't seen any delayed non-ICANS neurotoxicity. So no patients so far have developed cranial nerve palsies or Parkinsonism or Guillain-Barré syndrome. These things that we worry about that are rare but can occur so far haven't been reported for this study or for the phase 1 study, which is also being updated at this meeting, which has a much longer follow-up of 38 months. So there's 150 patients who've been treated so far with this product, and we haven't seen any of these events so far, so that's certainly very reassuring. As we would normally manage patients is how we would manage these side effects. So patients who develop cytokine release syndrome were treated in line with the protocol, as we would normally with anti-inflammatory medicines like tocilizumab if needed, or dexamethasone. Patients who developed early cytokine release syndrome within the first 2 days, generally was recommended to receive both of those agents. And as I said, only 2/3 of patients only had 1 dose of dex with their tocilizumab. Otherwise, things like infection. So we routinely prophylax patients when they get immunosuppressed. And so, all patients received immune prophylaxis with medicines to help prevent infection. And then infections as they occurred were treated, as we would treat any infection, in any patient that occurred post-CAR T. In terms of the ICANS, again, that was all managed in line with what we would normally have considered to be standard practice. So this is an exciting time for patients. I often tell my patients that this is probably the best time to have myeloma that it's ever been. And we're seeing a lot of amazing treatments come through with very deep and very impressive responses. And really what patients want is they want to get the best response, the deepest response that keeps their disease well controlled or undetectable for as long as possible. And in an ideal world, and what's exciting for me at least about CAR T-cell therapy is that is in the absence of continuous treatment, that they come, they have their CAR T-cell therapy, and then they're off treatment. They're not on continuous delivered medicines. So I think that it's very exciting to see these very deep responses. It's an early data cut point, but certainly we're going to see longer follow-up as the trial matures. And there's a new study that's going to be enrolling around the world, the iMMagine study, which is going to be enrolling patients who've had even less prior treatment, so 1 to 3 prior lines of treatment. So that's going to be very exciting for patients to participate in. So again, we'll have to see more mature follow-up, extended results, the whole trial population, and then certainly the results of the iMMagine study, which is now enrolling, which is comparing anito-cel to standard of care regimens. All of these things will be required to read out before we'll be able to have it in our hands.